So for this video, we're going to talk about glycosylation. This was the predominantly most often referenced concept from the Reflections of the Sugar lecture that was confusing. And most of you said the same thing, which was that you didn't feel like glycosylation itself was all that confusing. It was more that at that end of the lecture, we kind of sped up, as I know I often do, and so it was gone over a little too quickly. So rather than try to explain it differently, I'm just going to give glycosylation the time that it probably needed just to be understood. And of course, with glycosylation comes the idea of glycoproteins, and glycoproteins lead us to lectins. So we'll kind of just tell that whole story as one continuous thought. So I won't go too much into this, but a protein is made through transcription and translation of a gene, transcription occurring in the nucleus and translation occurring, in this case, in the endoplasmic reticulum, the rough endoplasmic reticulum. And the first place that protein goes after it's made is to the Golgi complex, to the Golgi. And in the Golgi, something called post-translational modifications occur to proteins. And that's exactly what it sounds like. These are changes to the protein that are made after translation, after the protein is synthesized by the ribosome. So one of the most common post-translational modifications that occurs in the Golgi is the addition of a sugar tree. Now, I'm not drawing this sugar tree exactly as you would find it. I don't have it memorized, nor would you have to. But this sugar tree is pretty much made up of two types of sugars. N-acetylglucosamine, which is a derivative of glucose. It's a monosaccharide, it's a sugar. And mannose sugar. So you create this mannose sugar and N-acetylglucosamine sugar tree, and it really is drawn this way. It's a branched tree, branched the way that we talked about branching sugars in the lecture, branched the way that amylopectin is branched. And so we have this small tree pre-made in the Golgi complex. And so the first thing that happens in the Golgi complex is that this tree is added to the protein. That's one of the post-translational modifications. And I'm trying my hardest to both talk and draw this crazy starter sugar tree just as I've drawn it below. And hopefully I'm doing a reasonable job. You'll see that I did refer to this as a starter tree. And the reason why this is called a starter sugar tree is because once this tree is placed on a protein, additional sugars of different kinds are added to that tree. So every glycosylated protein, and that's what we're making here, every glycosylated protein starts with the same basic starter tree, but different sugars are added to that tree depending on what that protein is being glycosylated for. And some sugars get more than one tree on them. I'm going to be a little bit lazier here and draw this tree a little bit quicker. So what we have just made is a proteoglycan, a glycoprotein. We've made a protein that is largely protein with some sugars on it. A proteoglycan is largely sugar with a few small peptides on it. But this is a glycoprotein. It's a large protein, fully folded into its three-dimensional shape, and that protein has had sugars added to it in the Golgi. Now that protein leaves the Golgi, it either exists at the cell surface. If it has been exported to the cell membrane, it's going to be displayed on the cell surface, or it is secreted. And let's make this protein a cell membrane protein. So I'm going to draw here a cell membrane. And we're going to embed this protein into the cell membrane. We'll make it a transmembrane protein, just as we've described it. And it has that sugar tree. Again, imagine that these trees are identical to the ones that I had in the larger drawing. There's just no way I can possibly remember exactly as I've drawn it. And those sugar trees are always displayed outward facing, so they're always in the extracellular space on the outside of the cell. And for those of you who had cell and molecular biology, the way that this protein got there was that it became embedded in a vesicle. That vesicle was shipped to the cell membrane because of the adaptin proteins that brought the cargo receptors together. Dynamin kind of pinched the neck. Those of you who haven't had cell and molecular biology yet or who will not take it, don't worry about that material. I'm just trying to link concepts here. One way or another, this protein is exported to the cell membrane, and it becomes embedded in the cell membrane there as I've drawn it, with, again, the sugars displaying outward. So let's say that this is a white blood cell, and it's circulating in the cell in the bloodstream. And let's also say for a moment that this white blood cell is due to be recruited to a wound, to a site of a lesion on the skin. So the individual who's carrying this cell has experienced a cut. That cut has triggered an immune response. Interleukins and other chemicals have been sent throughout the bloodstream that an injury has occurred. And now this white blood cell needs to be recruited to that wound site through the blood. Of course, if it was recruited to that wound site, but had no way to stick to the wound, that blood would just carry it away as well. So we need some anchoring mechanism. We need some way for this white blood cell expressing this glycoprotein to be permanently, or at least semi-permanently, attached to its recruitment site. And that semi-permanent attachment occurs through membrane-bound proteins of other cells called lectins. So this protein is a lectin. Lectins are groups of proteins that specifically bind to and stick to glycoproteins. So this green lectin I've drawn here, I've tried to symbolize that it is specific to and binds to the glycoprotein that is in the white blood cell. This interaction now anchors and locks that white blood cell to the wound site. There's a subclass of lectins that is specific to cell-cell adhesion only, called selectins. Selectins are, again, membrane-bound proteins that specifically bind to glycoproteins, but they do so for cell-cell adhesion. So this hypothetical situation that I'm presenting to you now, where white blood cells are sticking to a wound site and being recruited to an injury, that is due to the action of a specific selectin, a member of the larger protein family of lectins. Additionally, zygotes, or very small embryos in early stage of development that are just made up of a handful of cells, stick to the uterine cells of the uterine lining through glycoprotein-selectin interactions, just as I've drawn here. So whenever one cell, often when one cell needs to stick to another, that mechanism of cell-cell adhesion is occurring due to a glycoprotein being expressed by one cell and a selectin being expressed by the other. And then the specific glycoprotein-selectin interaction that is occurring at the molecular level is the anchor by which these two cells stick to one another. That's pretty much the sum total of what we were trying to get at the end of that lecture. Again, I know I didn't say anything quite differently, but I do get the distinct impression from most of your reflections that it wasn't the content as much as it was the speed. So hopefully this was a little bit slower, a little bit more in context, and a little bit more deliberate to just emphasize the function of glycoproteins once they're made in the cell, and the primary function of glycoproteins, at least as we are concerned with lectins, is for one cell to stick to another due to the complementary interaction between glycoproteins, specifically the sugars they express, and lectins, specifically selectins, which interact with those sugars. So I certainly hope this helped.